a year to plan every year. I got a pirate hat this time, though. A nice one.
That's good. Ooh, it's very nice.
Pretty.
Very nice.
It's embossed? No. What's the word for that?
Ceaseless. I have made us live. Okay.
Ooh.
Okay.
No, it's called the thing. But my ear thing's gonna fall off.
Nous vivons.
We are going to do show, no? It's good show. We do. We talk science as we do. You know.
C'est l'heure de science.
I like science. Science is good. You guys ready for a show?
Ouais.
Yeah.
Because it's time for us to start the show.
Oh, fantastique.
We start the show for my babies. All my babies. My science babies. 3, 2. This is TWIS. This Week in Science, episode number 642, recorded on Wednesday, October 25th, 2017. Spooky science on TWISoween.
Woo!
Hey everyone, I am Dr. Kiki, and today we are going to fill your heads with Frankenpigs, shrunken heads, and vampires of sorts. But first...
Disclaimer, disclaimer, disclaimer. As darkness falls across the land, the science hour is close at hand. Creatures crawl in search of studies to analyze all that research unmuddies. And whichever stories shall be found will fill the airwaves with such a sound. Antimatter CRISPR pigs, jumping spiders, shrunken heads, medieval leprosy, spider squirrels, spread by squirrels, give evolved microbes, spider venom a whirl, radioactive zombie plastic tombs, robotic bees you'll hear real soon. And though you might not hear it live, your mind will shiver. Hope you survive, for no mere mortal can resist the science news here on TWIS. This Week in Science, Halloween edition, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Our Avasti minions, it be This Week in Science.
It is. There she blows.
I wouldn't keep taking my hat.
For my
Isis
babies.
Is me. Good science to Kiki and Blair.
Good science to all my sisters. My sisters, I do the science for my science babies. Yeah, nobody knows what my character is. Unless, of course, they watch Orphan Black. If you watch Orphan Black, you have some concept. It's not perfect. It'll get there before Halloween. It is pre-Halloween. And Blair, who did you bring to the show? Who are you this evening?
Je suis un chef de cuisine.
Oh ouais, tu fais le nourriture pour le manger, ouais.
Oui.
J'aime beaucoup les I can't think of anything.
Le fromage.
Le fromage. How I love le fromage. Ouais. It's so good. Justin's a pirate, as I'm sure you.
No,
you're not a pirate.
I thought I was a Minuteman. Did I mess this up again?
You arged. You arged.
I must have been kind of hacked. Didn't the Minutemen arg? I don't know.
I was using the evidence.
Reddit history.
Arg. All right, everyone. Welcome to another show. We have all sorts of science ahead for you. Some of it's scary. I mean, it all depends on your perspective. I don't think any of it's scary, but we are here. I have stories about Frankenpigs, bacteria doing what they want, doing what they do, and also something we just don't understand. It's a mystery.
What?
Justin, what do you have for us?
I've got radioactive zombie plastic, medieval leprosy squirrels, and why a wandering mind is a good thing to have. Oh, also RoboBees.
I really don't want any medieval leprosy squirrels.
No, this is my worst nightmare. This really is Halloween.
I know. Squirrels, Blair. Leprosy squirrels. And Blair, what is in the animal corner?
I brought spiders and shrunken heads and also, let's see, virtual reality pain.
Ugh. Yeah, the future's not so bright, is it, everybody? Or it might be. I don't know. Anywho, we've got a lot of stories coming up, everyone. TWISoween is in full effect. And as we jump into our new favorite segment of the show, I want to remind everyone that they can subscribe to the TWIS podcast on iTunes, on Google Play's podcast portal, Stitcher, Spreaker, TuneIn, wherever you find fantastic podcasts around the world, you will find us. You can also find us searching on YouTube and Facebook. Just look for This Week in Science. You can also go straight to the source at TWIS.org. We also, at TWIS.org, have our 2018 calendar available for pre-ordering. So TWIS.org, we got calendars. Blair drew them. They're so cute and amazing. It's a coloring calendar. I can't wait. So anyway, let's jump into This Week in What Has Science Done For Me Lately? This one's a short one, but it is absolutely true. Minion Selden McCabe wrote in to say, it allowed me to share my experience at Eclipse Totality with my online friends. And he shared the link to a video, which I will share on our webpage and in our description notes on YouTube and Facebook and elsewhere, so that you too can experience the awe and wonder and the joy of the eclipse that Selden McCabe and his friends experienced at Totality. It's true. I didn't, Blair, you and I didn't really do much of this, the live streaming or the recording. I put my camera away at Totality. Yeah.
I have my whole life to see pictures of eclipses, and this was my one potentially only chance to see it in real life. And I'm really thankful to people that took the time to do it, but I really didn't want a screen in between me and what was happening.
I figure there's going to be plenty of versions of that to see.
Yeah, but the thing is, there are versions of it. Everyone who did take video of those 2 minutes-ish of the eclipse this past summer, everyone who did was able to share it with people around the world where the eclipse was not happening.
Yes.
So it took something that was a very unique local experience and turned it into a global one.
Absolutely, yeah. And I know that at the zoo they had tried to have an eclipse viewing party, but it was fogged out. They couldn't see anything. So instead, they live streamed footage of the eclipse happening, and they were still able to participate.
And additionally, thank you, science, physics, cosmology, astrophysics, for letting us know how a heliocentric solar system works, and a spherical or oblate spheroid body such as the Earth, how it might rotate in space, canted at an angle towards the sun, with its own satellite moving around that could occasionally, in a predictable manner, block the view of the sun. So, hey, science, for being able to predict where we should be to see that. That is science. There we go.
Yeah.
All right, everyone. Remember, we need to keep filling this segment of the show up with your stories, your ideas about what is important about what science does for you. What has it done for you lately? Leave us a message on our Facebook page, facebook.com/thisweekinscience, and I will schedule it to be read. You know what? There's a limerick coming up in a few weeks.
Oh, boy.
Someone wrote a limerick.
That's exciting.
Very excited.
Someone listened to me.
Yes.
Hooray!
Someone
The greatest present of all.
The challenge was accepted, Blair.
More. Send more.
Yes, please send more, everyone. We are waiting for your stories to fill this part of the show. And let's move into the science. So, should I get just you want Franken pigs or do you want a mystery?
Ooh, I like them both. Is
I
there
want
a
the
Franken
pigs.
pig?
Give me the pigs.
Okay.
The chef is demanding more pigs in the kitchen.
The chef wants more piggies. I wonder why. So, let's talk about how these piggies may affect our cuisine coming up in the future. If Americans can get around the GMO aspect of this, in fact. However, researchers publishing in the Proceedings of the National Academy of Sciences used CRISPR-Cas9 to help pigs lose weight. And
the chef does not look pleased with the skinny pigs.
The look on Blair's face is like, wait, what?
That seems like the exact opposite of what we would want pigs to do.
Right. So we like the nice fatty pigs, the fat marbling the meat because it adds to the flavor for those people who are not vegetarians and do eat the bacon and the ham and all the things that come from the pigs. However, this research, they don't think that they're affecting fat deposition within muscle. What they were focusing on is specifically a particular protein called uncoupling protein 1 or UCP1. And this is responsible for something I've talked about before on the show called brown adipose tissue mediated thermogenesis. So we have two types of fat in our bodies. We have white fat and we have brown fat. White fat is for energy storage. Brown fat can actually burn the energy inside of it and use it as a form of heat for thermoregulation. So pigs, I did not know this, pigs have a problem thermoregulating. They don't keep themselves warm when it's cold, which is why they lay down so much fat. So pigs get really fat. They have a big layer of white fat because their bodies don't do really good at this natural heat production. And so they insulate instead. And farmers, ranchers who ranch pigs actually have to invest in cold climates in heating to keep their pigs warm. So there's an energy cost, an energy outlay in order to ranch pigs normally. So these researchers thought, well, what if we could adjust or insert actually this UCP1 gene into the pig and get it working so that the pigs could thermoregulate? How could this affect pig ranching down the line? And so they gave it a shot. They used CRISPR-Cas9 and they did this. They inserted mouse UCP1 into the UCP1 locus in the pigs, the porcine endogenous UCP1 locus. And the resultant pigs had a better ability for thermoregulation. They had less fat on their bodies, and they had an increased lean percentage. So more lean muscle. So hoping, I guess everyone's hoping that it won't affect the flavor of the meat and the marbling and the meat and the fat that gets into the meat itself and make that too lean. But in the long run, this could be something that is a big jump forward for the pig ranching industry.
That's interesting. You wonder what the cost benefit is if you lose weight on the pigs. So you lose money from the individual pigs for meat, but you're gaining money from not having to have the heater on.
Mm-hmm.
I wonder which would actually win out.
As much.
Yeah.
As much.
You know,
We'll have a bunch of dead pigs in the morning if we just leave the heat off. We gotta, how do you know how much, if your pigs are happy? You're gonna have to.
Who needs the fatty pigs anyway, huh? Yeah, so in this, it's pretty interesting. They the average, so according to the study, the fat was about 15% versus 20% in unmodified control. So it only lost about 5% of their fat. The average percentage of lean meat increased 3% from about 50% to 53%. So it's not significant. These were happy little pigs. Happy little piglets.
Happy little pigs becoming happy little bacon.
Someday. More bacon. CRISPR bacon. Crispier bacon? Yes, there's something there. I know. Okay, now moving on to the mystery. This is a mystery of the universe. We've had interviews before, we've talked before. And in fact, earlier this year, I talked about a study that was investigating the differences between the proton and the antiproton. Now, the reason somebody would look, a physicist would look at the differences between a proton and an antiproton is that we have a universe made up of matter. There's antimatter too, but when antimatter and matter connect, when they bump into each other, they annihilate. So, looking so far, there don't seem to be, there haven't been any differences found in between antimatter and matter. And so people have been wondering, they're like, how come we have a universe? How is it even possible that we have a universe? Why didn't it annihilate? There was asymmetry somewhere in the very beginning that allowed matter to dominate, and we still don't know why. And so researchers are still trying to figure out step by step what happened. So physicists at CERN in Switzerland, looking again at the proton and the antiproton, they trapped antiprotons within a magnetic field and held them there. It's a special kind of trap for antimatter. It's called a penning trap. It uses magnetic and electric fields to hold these particles in place so they don't touch anything, because if they touch something, they get destroyed, right?
They've lost their study material. Yeah.
Yeah. So they have to trap these things. And so they trap them in the penning traps, and they used 2 traps to make the most perfect antimatter chamber ever. And they held the antiprotons for 405 days. That's over a year. Yeah. They trapped these antiprotons for over a year. And they measured and measured and measured, trying to find something that's called the magnetic moment. But in the case of the antimatter, the antiproton, it's the anti-magnetic moment. And this is the number that measures how this proton, this particle, reacts to a magnetic force, how strongly attracted it is to a magnetic force. And so they've measured this for protons previously. And now they measured it for antiprotons. They have a value of minus 2.79284734441 micronewtons, or it's a nuclear magneton. A nuclear magneton. Minus 2.79. Anyway, it's negative. That's the only difference from the proton. It matches exactly. The way that the antiproton is attracted or reacts to a magnetic force is exactly the same, but opposite to a proton. Huh. So still, we have no idea why the universe is here. Still. Over a year of testing, almost a year and a third of testing, and no clue, no idea. And so studies are going to be booted up at Alpha at CERN. It's another experimental chamber. And they're going to be studying gravity and antimatter to see what kind of effects gravity have. And, if gravity pulls matter down or toward it, will antimatter be repelled? Does antimatter fall up?
Yikes.
Yeah.
Magnetism, but not maybe gravity. But I do love I love the title of this so much. The title of this paper, Universe Shouldn't Exist, CERN physicists conclude.
Yep.
That's just...
CERN
We shouldn't exist.
We shouldn't exist. Well, it's not that we shouldn't exist. It's just we don't know what makes the difference at this point in time. We haven't figured out how matter dominated. If all the measurements up to this point, we haven't made all the measurements yet. That's the thing. We have to make all the measurements to figure out what is the factor, what parameter leads to asymmetry. And that's what we need to figure out. We don't know. Nothing related to the physical particles of antimatter at this point in time have us coming up with any conclusions. So it's a mystery.
We shouldn't exist. Be sure to tell children that on this chilling Halloween.
That's right.
Children, the rules of the universe tell us we shouldn't exist.
You shouldn't be here.
So think about that when you're going to bed, small child. Why can't you sleep? Because we shouldn't exist.
Or stony person. Anyway.
Huh?
This is This Week in Science. Justin, what did you bring?
Reported in the Journal of Medical Microbiology. They identified a strain of Mycobacterium leprae, the bacteria that causes leprosy, and ancient DNA extracted from a female skull discovered in England around Suffolk area. Through radiocarbon dating, a team of researchers confirmed that the woman lived between 885 and 1015 AD. They took shavings of bone from the skull in order to extract the DNA, and they also there detected the bacteria that causes leprosy. The woman from Hoxne is one of a growing number of medieval leprosy cases identified in human remains found in and around East Anglia in the early medieval or Norman-ish period. Researchers suggest an explanation for the prevalence of leprosy in the area may be found in the medieval trade, possibly fur, which would have included one animal known to carry the disease even to this day: squirrels.
You just made Blair so happy.
Yeah.
No, it's just proving my point. They can't be trusted.
No. There may be good reason. There may be good historical reason, Blair, for us to be wary of squirrels.
See?
Right?
What have I been telling you all?
This is disfiguring disease is likely to have had a severe impact on this particular woman. They could find all sorts of visible damage to the skull. She probably had extensive facial lesions and is likely to have suffered nerve damage all the way out to her extremities. Analysis of the bacteria revealed that the woman from Hoxne had been infected with the same strain of leprosy already identified in skeletal remains found from a man in Great Chesterford, who lived as early as 415 to 545 AD, suggesting that the leprosy had persisted for hundreds of years in the southeast of Britain. Sarah Inskip, research associate at St. John's College, Cambridge, lead author of the paper, said, "This new evidence, coupled with the prevalence of leper hospitals in East Anglia from the 11th century onwards, adds weight to the idea that the disease was endemic to the region earlier than other parts of the country. Same strain of leprosy has also been identified in skeletal remains of medieval Denmark and Sweden." And the study authors suggest North Sea trade links with Scandinavia may offer an explanation for the apparent prevalence of the disease in both places. It's possible that this strain of leprosy was proliferated in the southeast of England by contact with highly prized squirrel pelt and meat, which was traded by the Vikings at the time this woman was alive. So it's one of those things you don't know about Vikings. We're really into squirrel meat, apparently.
Yeah.
So there's easy to catch. Squirrels are prolific.
Mm-hmm.
Squirrels like nuts. I don't know.
Well, last case of human leprosy, in case you're afraid of going to the UK now, in the British Isles, this was over 200 years ago. But a recent study, this is like just a couple of years ago, demonstrated that leprosy infection in red squirrels and brown sea squirrels in island Dorset persists to this day. And the disease that affects those squirrels is very closely related to that detected from the woman from Hoxne. So,
see, so what did we learn from this? Stay away from squirrels.
Yeah, it's the same strain that infects only one other animal-based source of this is known to exist, and that is
Armadillos.
That's it. The 9-banded armadillo, which has caused leprosy cases in Florida.
Hashtag fun fact.
Never heard. Yeah, right. Oh my gosh. So one of the things in terms of the Denmark and Sweden cases, it is questionable. This is in Skip again. It is questionable how long the bacteria could have survived on fur or meat, but it's notable that squirrels were also sometimes kept as pets.
Barf.
Squirrels are smart. I can see how that can happen. They're cute and fluffy and
pet squirrels.
Yeah, I mean, if you're coming across, as Blair has said, if you come across a squirrel in a park, just really tamp down that urge to feed the squirrels.
Don't feed wild animals.
Don't feed wild animals.
Here's one of the reasons why.
Not advisable.
I get a leash on my daughter. She chases squirrels. She's like, she sees a squirrel, we'll chase it and right up the tree.
And sometimes squirrels turn around and chase you back. For real.
She'd probably just try to catch it. So, but now that I know she can get leprosy from a squirrel, who knew?
Tamp that down.
Yeah.
But that's in England, not here, right?
Well, I don't know. I haven't looked for it. So, but I'm going to assume all squirrels cause leprosy from now on. It's just a safe way to do it.
They can get you sick, whether it's leprosy or not. They could carry other things too. So it's just a good idea
not to
feed wild animals that are not super scared of humans that have sharp, pointy teeth.
Ed from Connecticut says that his grandfather ate squirrel stew. And roadkill in rural Maine is how they shop for meat. Ew. Yeah.
Yikes.
Yeah, leave the roadkill alone, people. It's not a federal offense.
Yeah.
Yeah, don't mess with the roadkill.
Don't mess with the roadkill. Don't feed the squirrels.
Don't get leprosy from the squirrels by not messing with the squirrels. There we go. Yeah. This is This Week in Science. And do you know what time it is?
What time is it?
It's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. That
What you got, Blair?
Ooh, I have shrunken heads. Not the kind that you would find in a voodoo shop, but instead the shrinking
secondhand store.
That's right.
No.
Yeah, there's a whole song about that, isn't there? Witch doctor. And anyway, red-toothed shrews. Red-toothed shrews, while alive, have heads that shrink. This is like an actual individual animal whose entire head shrinks, gets large again, and shrinks again as a seasonal adaptation to winter.
I can do that. Watch.
This is not a teeny tiny amount of shrinking. This is 20%.
What?
Yes.
Wait a second.
Yes. So this all started with an early study looking at the population level. So they were just catching random shrews, measuring their brain case. They found that the height of the brain case declined by 20% and then increased again in the spring by 15%. They created a name for this weird phenomenon after the first person who reported the observation, the Denel phenomenon, Dehnel. But this new study, which is from the Max Planck Institute for Ornithology in Germany, which is
because
interesting.
birds. No, the thing is, no. This is part of the work that I did in grad school.
Oh, let's hear it.
Birds' hippocampuses grow during the fall. So they birds that migrate, and birds that store food have changes in regions of their brain.
But then does it? So it grows. Does it shrink again?
It does. Yes. A bird that stores food in the end of the summer and the fall, the brain starts growing and the bird starts storing food.
Okay.
And then they survive on that food over the winter and into the spring. And then it's mating season and their brains shrink.
Okay. So this is not so bizarre. I was reading the study and
this is mammals. This is mammals.
Losing my mind.
Okay.
Because so it all started with this original study looking at the population level, just grabbing random shrews. This new study actually takes specific shrews. They follow individuals over the course of seasons. So from the summer of 2014 into fall of 2015, they captured a shrew, they anesthetized it, they x-rayed their skull and implanted a microchip. So this was not just brain case size. They were really looking at the size of their brain, the size of their skull, everything. And they found that the head shrank over the course of the season. And then they also showed regrowth in spring. They are still not sure what the cause of this is. They also found that their entire body shrinks in the winter. Their spine gets shorter, their organs lose mass, but their brain mass is the thing that decreases by 20 to 30%. Imagine losing a third of your brain. I can't imagine. So shrews don't migrate or hibernate in winter, but they live in some pretty cold places. So the idea is because they have high metabolisms, they have a very high energy demand, that their decline in brain size actually helps them need less food because the brain is so energetic. I think what I see is actually a bit of a feedback loop, because what does your body do in starvation? It starts to eat its own organs, right? So potentially their body is burning this stuff for energy and at the same time reducing the need for more energy because they've burned brain. So that would be my hypothesis, is that it's actually kind of a 1-2 punch. But they are saying that it looks like the evidence points to the fact that the cranial structures are reabsorbed and then they regenerate bone tissue. It just seems so crazy. But this is every single individual that they measured this happened to. This was not something that it was just the bigger ones. Right. That was the original explanation, was that because of population dynamics, it was actually the big ones were dying, so the average decreased. No, it's every single shrew. Their brain shrinks. Their entire head shrinks.
That's wild. And it's also wild because it's such a strange thing. And why have we not seen this before?
Why have we not seen this before? And what is happening?
Nobody was measuring it. I mean, it's just, all it is asking the right question.
Well, because these shrews are like this big. They're like smaller than the palm of your hand. So you could see how an untrained eye, it's not like elephant heads are shrinking by 20%. That would be very obvious, right?
We would think we would notice.
This little fuzzy shrew that hides underground most of the time anyway, of course it's going unnoticed if we're not measuring it. What I would be very interested to see is if it is just a product of starvation, or if there is a hormone trigger that makes it start happening before they actually start starving. And if you stopped it, what would happen to their body? Would their brain grow each spring if it didn't shrink in the winter?
So this was a field study, right? This is a field study. So they're retrapping individuals over seasons, measuring them, putting them back in the wild, and seeing, who lasted what anyway.
Yeah.
What you're suggesting doing is starving shrews. For science. Bringing shrews into the lab. This is the study that needs to be done. You bring shrews into the lab, and you basically have a year-long study Some of the animals are on a regular schedule with normal food. Others get food deprivation springtime. Others get food deprivation in fall.
Right.
There you go. That's
Absolutely.
what they need to do.
Yeah,
Yes.
and what the researchers
and then they can follow hormone levels through the entire time. Yeah.
Yeah, and what the researchers want to do next, which is not what I suggested, is they actually want to know if the process affects cognitive abilities, which is a great question. And they also want to know structurally in the brain what happens as the head shrinks. So is it the same in every shrew, or is it different? What part of the brain gets eaten up? These are great questions. But this bizarre phenomenon has huge avenues of research just asking to be explored.
And it's pretty bizarre, but we've also seen this and reported on this in dinosaurs, where we weren't looking at brain size, obviously, but that part of the problem with morphology was that you were identifying dinosaurs based on cranial formations. And what they found was a lot of these different species turned out to be the same species that in a lifetime will grow a bit of skull or horny bit here and then reabsorb it, and it will be gone as they get older. And so there have been some pretty interesting strategies in formation of skull over the years. I haven't seen any one of these strategies survive.
Yeah.
Until we get to this shrew.
Right.
That's actually changing morphology.
So just try to bring this to an animal that you can kind of think of a little more, you have more familiarity with, like a dog. Imagine your dog, human, a quarter to a third of their head size gone. Their head shrinks down, but their body also shrinks a little bit, but not as much as their head. And then it comes back every year.
Yeah. I was already just picturing this in people, like how.
Yeah, well, that's a whole nother barrel of fish. But yeah, a pretty weird stuff, shrunken heads. Yeah. And then moving on to spiders.
I like spiders.
It wouldn't be.
Some people think they're scary.
Yes, it's true. But it wouldn't be Blair's Animal Corner without some spiders on Halloween. But I particularly, want to talk about this guy. That's not scary. Look at this little fun lady. This is a jumping spider, the cutest of the spiders. I just want macro pictures of jumping spiders all over my home someday. If I
with their eyes staring at you in the night.
If I have kids and their friends come over, they'll just be like, where are we? Anyway, so jumping spiders. So jumping spiders are, of course, famous for mating displays. The peacock jumping spider, of course, the amazing dance that they do. The males have to make a pretty big display for the female to acquiesce, to actually go through mating. Well, the plot thickens with jumping spider mating because their strategies on the female side are kind of extreme. And that is that they actually will only mate once in their entire life. And this is not a thing where, the males, they have terminal copulation where they lose their...
Oh no, you're breaking up. Where'd Blair's internet go, or is that me?
No, it's
Blair broke.
Oh, you're back.
I'm back though. This is important.
So you're right at the point. You're right at the point where they were losing. It's not that the male spiders are losing their.
So they lose their copulatory organ, right? That's one.
Some spiders.
Some spiders. The females eat the male after copulation. That's a one and done scenario.
Not this spider though.
Not this one. This one, the female does it once, and she goes, "I'm good. I'm good for life." After the one time. So let's explore this for a second. So in other invertebrates, a lot of the time they go through sperm competition inside the female. So if the female has a mating event multiple times during her life, then there will be sperm from different males in her body, and that actually has an internal competition for which sperm will win and actually help make a baby. Then the other option is, males have special organs to scoop out sperm so that they can displace it with new sperm. There's all these sorts of things because in invertebrates, a lot of time, sperm can last a lifetime. It doesn't really have a shelf life for them. So they can actually they can retain that sperm, the female, for multiple generations of babies. So because of that, it would appear that the jumping spider, she's good with one time. So the really interesting part would be
so she's not just putting out one is it brood of spiders? She can do this later too.
You can have a whole lifetime, multiple generations of babies. She's good on one mating event. So this was a study in Australia looking at 89 female jumping spiders, Cervea incana, and they were caught as immature spiders in Australia. So they were virgin, and they were brought into the laboratory. Each female was paired with a different male daily for the first 10 days of her life, of her adult life, after, she hit spider puberty. And then every 10 days thereafter, for the rest of her life, they switched males. And they found that the first mating induced sexual inhibition in most of the female jumping spiders. So the most common method was to mate only once in their lifetime. A few of them mated twice. Almost nobody mated more than twice in their lifetime. After mating, females were so unreceptive to other males that they would fend them off with their legs and turn away. They would push them away. And so, yeah.
I don't need you.
Exactly. She goes, I got some. I'm good. So they can store their sperm for the rest of their lives, like I said. So this is interesting because whoever gets to the female, the virgin female, wins the reproduction game. That's it. Because that female, after the first event, she's done. So
there's no sperm competition. There's nothing. She's storing it and she's keeping it.
So the amount of competition between males for virgin females in jumping spiders has got to be insane, which totally changes the way we look at jumping spider evolution too, because this means that one male can monopolize a female just by getting to her first.
But this is just the Australian jumping spider.
Right.
This isn't necessarily all jumping spiders.
Right. So this is this one Cervea incana jumping spider. So so that's the case with them. So now it's time to turn to other jumping spiders, see if this is actually happening. What the other thing that would be really interesting to study based on this is why. Is there, again, a hormonal change in the spider that's making them say, no, thank you? Or is it just that they have enough? They have kind of a reserve. It's been filled. They're good. So the fact also then that the females can fend off the males, which I guess because they're spiders, generally speaking, the females are a little bit better. They have a little bit more of an upper hand in the mating game than in other species. In other species, males can kind of push their agenda, whether the female's interested or not. But in most spider species, the females can kind of call the shots in mating. So
yeah, very often in spiders, they're either equal size or the female is larger, right?
Yeah. Yeah. And jumping spiders, usually they're the same size, but the females are more aggressive. So if you ever see the peacock spider videos, if you ever watch those, the males, when they're dancing, eventually if the female's not interested, she'll actually turn and jump at the male and the male will run away because the female is more aggressive. So anyway, the kind of mold breaking element to this story is the fact that sperm competition is not part of this. The males cannot display to convince a female who's already been mated to mate. That is really a one and done scenario. So how do the males compete for virgin males? And then how has evolution not caught up here and figured out a way for males to be able to sneak in and get more matings out of this deal? And will that happen soon? That's the other thing, you know, like you talk about evolutionary arms races with predator and prey, but I think about it a lot in terms of male and female, because a lot of the time, you know, the males and the females of a species have different concerns, different investments, different priorities with mating. And so there is kind of an arms race of trying to figure out who's going to call the shots in a mating event. And so I think it's fascinating. It's unlike anything I've heard of. And I can't wait to see kind of what the mechanism for this choice from the female is and hear about more of the background here.
Yeah, it's almost like I wonder if as a strategy, you know, how good that is. I mean, obviously jumping spiders survived to the modern day, so it's survived all of its evolutionary history. Then I'm wondering, is this like a new thing? Is this going to last? Because there could be some risk of getting a shallower sort of gene pool, like sort of the point of the multi-partner or the sperm competition is that you're mixing up that genetic soup so that you're giving enough looks at different things to out of the different next generations. There's enough variety that they may be better if equipped in some directions, maybe a little less in others. So it's a spreading of your chances and your odds of survival. And it seems like a very possibly narrowing of a gene pool. But then again, how many are in a spider's brood? Like there's probably a lot of spiders, and so just by that sheer number, it just didn't matter.
So
anymore.
As long as one male spider isn't inseminating all of the females in an area, it should be okay.
Which is very unlikely to happen.
Yeah.
All right, everybody. I think we've made it through the spooky first half of This Week in Science TWISoween. That's right. This is This Week in Science. We're going to take it to the break right now, and I hope everyone stays with us for a few moments for our messages. We will be back with more spooky science after this. My babies.
Explain the things you heard with more than intuition. The line of reason shows the way to go. No conclusion. The methods of hypothesis and patience are the only.
Hey, everybody. I do hope that you are enjoying the TWISoween edition of This Week in Science. We sure are having fun here on this end. Head over to twis.org, you guys. Because you know what's there? Pre-ordering for the 2018 Blair's Animal Corner This Week in Science calendar, which Blair has hand-drawn. It's black and white this year. Well, you know, she's colorblind. She's done a good job so far. But black and white, not because she's colorblind, but because it's a coloring calendar. You guys get to take your own artistic tendencies out on the calendar this year. And the calendar will be full of all the usual goodness, all the fun TWIS science holidays, all of the amazing news of what shows we're doing, basically every Wednesday. But it reminds you, so you know. And you know what was on this calendar? TWISoween. It told you that tonight was TWISoween, so you knew to look forward to it. If you were looking at your calendar from 2017, don't be stuck without it next year. Now is the time to pre-order. Head to twis.org and click on the pre-order link. Get yourself the 2018 TWIS Blair's Animal Corner calendar. It's going to be so fun, you guys. Get it, get it, get it. And then after you get yourself that calendar, if you're still interested in supporting TWIS and maybe thinking about your holiday shopping, because the holidays are coming up, head over, you're at twis.org, already. So why don't you just take a little gander at our Zazzle store while you're there? So all you really need to do, it's super easy. You click on the link that says Zazzle store. It's right there, little Zazzle store link. Click on it. It takes you to our Zazzle store, and you will be able to peruse all the offerings. We've got lumbar pillows. We've got tote bags. We've got T-shirts and mugs and hats and stamps. You can even get stamps for your holiday cards if you send those out. We have so many goodies with the TWIS logo emblazoned, and also with imagery from the last couple of Blair's Animal Corner calendars. The hand-drawn art is amazing. I mean, the woolly mammoth lumbar pillow is just my favorite. I love it so much. And you can share all this goodness with people you care about these holidays by shopping on the TWIS Zazzle store. It's that easy. Get somebody you love a tote bag from TWIS. Totes my goats. Additionally, if you're thinking about, hey, I just really want to give TWIS a present this year, you can do that by going to twis.org, clicking on our donate button. You just click on that donate button, and you will be able to donate whatever amount you would like through the PayPal interface that comes after. If you're interested in donating in an ongoing way, you can click the Patreon link at the top of the page. That takes you to our Patreon community, which is growing and amazing. The list of patrons I get to read every week at the end of the show gets longer every month. I am so thrilled about this. And you can become a patron simply by clicking on the red Become a Patron link. And then you can become a patron at whatever level you would like, and we will send you little goodies. We might even say your name on the air. It all depends. But you will be part of the community at any level, and you will be privy to little emails, little messages I send out, little videos occasionally, inside scoops through the Patreon community. So we do hope you join up there if you can. For those of you who are not interested in supporting TWIS in the financial way or in buying things, I know all this buying of stuff, right? If you're not into that, help us spread the word. Help us get the word out about TWIS. Share the most recent episode. Share the fact that you are watching us right now, listening to us right now. Share that on social media. Get the word out. Get other people watching and listening, because that'll grow TWIS, and it'll make everything we do that much better for you. You support us. You help us produce. You are our executive producers in that fashion. We thank you so much for your support. We really could not do this without you. My babies could not do this without you. My babies. My sisters. Where are my sisters?
I'm going to have to watch this show just
dollars and cents all these authors receive. If miracle wonders were held in their looks, why waste precious time and try selling their books? Why sit and wait for your publishing royalty? If one has real power, who needs reader loyalty? If you could travel by thought to a mystic.
And we're back with more This Week in Science.
Yeah, we are. Justin, what you got?
So in the intro to the show, I teased a story about radioactive zombie plastics. And now that I'm about to do the story, I realize I may have overstated the radioactive zombie bit.
It's just plastic.
And well, actually, there is plastic involved, but the story is about concrete. But concrete can be exciting to talk about as well. After all, concrete is the second most widely used material on the planet. Can you guess what the first is? Water, which is actually used in making concrete as well. It's the number one used material, and it's even used in the number two material. The manufacturing of concrete generates about 4.5% of the world's human-induced carbon dioxide emissions. What? 4.5%? That's a bigger number than I would have guesstimated. MIT undergraduates have found that by exposing plastic flakes to small harmless doses of gamma radiation, then pulverizing the flakes into a fine powder, they can mix the plastic with cement paste to produce concrete that is up to 20% stronger than conventional concrete. Yay, stronger concrete. But that's not all. Their admixture will adds up to 1.5% of the total material in the concrete, which, if utilized everywhere tomorrow, would reduce human output footprint of the world's carbon dioxide emissions by 0.0675%, which is a really big amount, despite being starting with a point followed by a 0 and then some numbers. It's a massive impact, really. This is a quote from Michael Short, assistant professor in MIT's Department of Nuclear Science and Engineering. There's a huge amount of plastic that is landfilled every year. Our technology takes plastic out of the landfill. Actually, they took no plastics out of landfills in the study. Actually, they got the plastics that they used from a recycling plant, and there's very little indication the invention itself would increase recycling efforts by individuals. Maybe, though, with a little industrial competition for recycled plastics, the reduce, reuse, recycle movement could get a little bit more support. This paper appears in the journal Waste Management.
It's interesting. I'd be a little worried. I'd want to do some testing on weathering on the concrete to see if plastic would end up in the water column from the concrete.
Yeah, it's in there. So actually, what happens?
That would be my concern.
But
what happens when they hit it with the?
We use recycled plastics.
Well, that's what they're using. Yeah, they're using recycled plastics.
And so then you're keeping them out of the landfill, putting it in the concrete, and then at least it'll take longer to end up in the water column.
Right. Yeah. So when they took this, what they did was, it's a good question. They used the Cobalt 60 radiator that emits gamma rays at MIT. It's something that's currently normally used to sort of decontaminate food. Right? They run it past this gamma ray thing. It's not something that leaves residual radiation. You're not going to have detectable radioactivity from this. But it changed the structure of the plastics. It made it it turned it into a more crystalline form that actually made more bonding connections with the concrete. So to your question about weathering, it's less likely to leak out of the concrete than any of the other ingredients of concrete because it's more bound. It's actually a little bit denser. One of the interesting things, or left less spaces. I don't know if its weight is any greater. But one of they tested it through an X-ray micromography machine, and they revealed that their radiated plastics, especially at the higher doses, exhibited these crystalline structures. There's more cross-linking, more molecular connections, which is what led to this sturdier concrete. And they put it through all kinds of other tests as well to see how strong it was. Let's see. They increased the strength. It was 20% compared to samples made with Portland cement. And they also did some another mixed cement, which it was also a little stronger with, which is what they used for their final formula, which is they use fly ash and silica fume, which is by itself a little bit stronger than Portland cement. So they ran it through some pretty good compression tests to see what it did. But this is
as long as they continue to use recycled plastics and they don't eventually find that they love this concrete so much that they need to produce plastic for it.
Yeah. It was a sort of sideways enjoyment of the story was the fact that they kept talking about how this could take plastics out of landfills, and then they talk about how they went to a recycling plant to get their plastic. And I was like, I guess it's harder to imagine bunch of MIT students rummaging around a landfill in search of the materials for their project. But I still don't see, maybe they just haven't been recycling at their lab and just like, hey, this has a use. We could still use this for something.
Well, there are a lot of bottles that do get thrown into the trash. And if they could be separated out because there's a financial impetus for it, then maybe that would happen. Secondarily, when you're doing plastic recycling, it's not cheap. That is a money intensive, heat intensive, it is an energy intensive process. And compared to say aluminum recycling, which is fairly straightforward, plastic is not easy. That's why they say certain types of plastic. You have to look for the special symbol on the bottom, and it has to be the certain type of recyclable plastic because it's amenable to the processes that we currently use. So it could be great. And if you could, and if this is the process of, hey, these things, they'd get recycled maybe into other plastic bottles, but maybe they would go into cement. Does it really matter?
Yes, as long as we don't make more plastic.
It's like, yeah, and at this point in time, if we stopped making plastic, is there enough plastic on the planet to support cement production?
Oh, yeah, totally. Yeah,
especially if we skim the oceans for our plastic.
I love this though. This is like these bottles are becoming this plastic is like the Spider-Man of concrete. It's like we put gamma rays on the plastic bottles, or what was it? The Hulk? Was the Hulk the gamma rays? Gamma radiation?
Yeah, I think it was.
So
yeah.
This is it. Plastic is the Hulk for concrete.
That's great.
Okay, so I had a couple of stories this week that I just thought were super interesting and I wanted to discuss.
Great.
Because evolution, it's so fascinating and there's so many questions about what's happening now in terms of climate change and human impact on environments and also where things are going to be going. Are we in the middle of the 6th mass extinction? Is it human caused? We don't know. So here we go. We're talking about evolution. The first story I ran across this week was on Vox.com, and it was with regards to a new book that is coming out. There's a conservation biologist who has written a book called Inheritors of the Earth: How Nature is Thriving in an Age of Extinction. And his premise, this Chris Thomas from the University of York in the UK, he says, yeah, okay, we're killing a lot of things. There's a lot of stuff dying right now. But as we kill things, other things are gaining ground and are making way and leading to speciation and diversification.
Wait, what?
Yes.
Well, yeah, because it's leaving empty areas in habitats.
Yes and no. So because of human activities, there's been increased hybridization because different species are being moved by humans, and also because of the way climate is being affected, species are moving into different areas, and the niches that they have existed in are overlapping more and more. And so there's hybridization that's occurring. There is also, there are new urban niches that have been created that were not available before because of humans using the environment. We have changed it and created new environments for wildlife to thrive. Pigeons, the house sparrow, among others. Squirrels with their leprosy, yes. Flies, various things. So there are new hybrids, chimeras, and just species that are becoming very prevalent that were not before. And so there's this interview on Vox between Ferris Jabber and Chris Thomas is fascinating to me because Chris Thomas doesn't deny that there is extinction happening. His premise, however, is that give it 10,000 years, the scale of evolution, the scale of change, the scale of extinctions and biodiversity booms is much larger than the 100 to 200 years that we are looking at right now. Look at it on the 10,000 year scale. And all the little debates that we're having about invasive species, this and that and the other, are completely moot. And species are going to be thriving because of humans.
Oh my gosh. So I love this argument because they're kind of right? Like the Earth will be here.
Yeah, it's not
No
going
matter
anywhere.
what we
Life
do,
abides.
Life abides, the earth will be here. But the question, that's not the question. That's never been the question to me. The question has been, what do you want it to look like here now? And for the next several hundred years, for our children, for our grandchildren, for our grandchildren's children. And do we want it to be filled with amazing species going extinct and intense climate change and deforestation? Or do we want to try to maintain some of the natural beauty that we have, right? So I'm totally on board with that, saying that we are altering the environment. And as we alter the environment, species will change and adapt. There will be new evolutionary radiation. That is just the way it works, right? That's the nature of evolution. But I think also you need to take a step back, and while that's a fine point, that's not really the argument. I would argue.
Right.
The argument is the argument he's making is just that the earth will rebound eventually. So it doesn't matter what sort of devastation we commit upon the earth now, because someday it'll go back to whatever it was. It's been, no big deal.
Fine. But what about us? What about now? What about these species? Like, that's the question: is what kind of impact do you want to have as a species on this planet?
It applies to everything. Global warming, so what? The earth will change climates again, 200,000 years from now, it'll be different, be fine. As if nothing that we do today is impacting us now. And that's the argument that he's making. It's not about what we do now, what we do. And who he's probably talking to is somebody who's not engaged with nature or doesn't know the food cycle or where their food comes from.
No.
I think so. I think they're saying, look, your day-to-day life isn't going to be affected no matter how much we pollute.
Well, that's not true.
Go away
or at all.
Go extinct.
Let me tell you some of his.
No,
his
it's not
thoughts.
true, but that's what's.
Yeah, his thoughts. One of the quotes from this interview, he says, in many respects, we are fighting the origination of new species. We are doing things that slow that down deliberately. In particular, I'm thinking of the fact that we usually take a disliking to new hybrids, think they're somehow impure, and try to exterminate them, when actually this is a natural process by which new genetic diversity is created. There's a great argument running through the global shift in conservation thinking toward ecosystem goods and services, where more biological diversity is in principle associated with more good things we get from the earth. Everything from personal enjoyment to conditioning of the atmosphere, retaining soils, keeping water clean. Almost all of these good things we get from biological diversity, we get from species quite close to us. And he says, I like the idea that there are giant pandas, Blair, in the forest in parts of China, but I don't benefit in terms of the direct ecosystem goods and services in and around myself from the fact that there's some rare species at the other end of the planet. The number of species in the region that you live in is much more relevant to the delivery of the good things that we get from nature. And that is the element of biodiversity. How many species in each region, that seems to be going up around the world.
Who is this person? Are they a scientist?
Yes.
Right. So they are making the point that I was saying. They are making that point that if you don't notice.
I have a real question.
It doesn't matter.
What kind of science?
Who is he? He is a conservation biology professor.
Wow. So this is astounding to me because that is like
antithetical to conservation biology, right?
Straight up false. Like, we know that the coral reefs in the tropics give us oxygen here. Yeah. We know that. There is science. You can find that. Like, these far off places have direct impacts on our daily life. The climate change at the tropics affects currents that affect our fish right off our coast. This is astounding to me. We just did a story recently about the ecosystem services of pandas, and that's, as much as, it irked me. They definitely did say that there were clear, measured impacts on the bamboo forest from the pandas. And the fact that we have medicine from the rainforests that are keeping people alive that are not biologically similar to us or geographically near us. This is fascinating to me that
would be the argument he takes, the locality. Yeah.
The broad brush that's being painted with is kind of surprising. Like at the start, I totally understood where he was coming from, which I do think it's important to like take that step back and also remember that this is the main reason we care about conservation is actually kind of selfish, right? Because no matter what, the earth will continue. So in some ways that actually makes people care who are not as much of environmentalists naturally, if you talk about how it actually affects their life, and it's a good investment for them personally to care about the environment right now. But so I was totally on board with him at the beginning.
I know his view is very interesting. It's actually very nuanced and very, very different from what we've heard from so much of conservation biology. And he actually says that mass extinctions, when we call them mass extinctions, that is 75% of you cull about 75% of species for it to be a mass extinction. Right now, we are somewhere on the order of 5% of mammal species to 10% of bird species in the last 50 to 100,000 years since humans
left. What about
left
invertebrates, though? And what
that's like there was a story last week that we talked before about the insects being lost in Germany. Yes, amphibians. So mammals, birds, that's one part, but you have to look at the entire picture. So yes, how close are we to that 75%? And he argues that there's actually a very high rate of species birth right now, and that we should be looking at that and not just bemoaning the fact that some species are dying.
And again, there's elements to this
change
that
is natural, right?
Like protecting new hybrids and not trying to exterminate hybrids. I totally understand where he's coming from with that. But I would argue that it's nowhere close to equal, the rate of extinction to the greatest
I'm
speciation.
I'm sure it's not.
Nowhere close.
Yeah. But the question is, if we're going to be trying to conserve species, do we want to conserve the past or do we want to conserve the future? And if we're going to take and this is an interesting point to take from his ideas, if we are going to be optimistic that there is going to be a biodiversity boom of sorts as a result of all the damage we're doing, all the things we're doing to our environment, not even think about it as damage, just the change we've affected. If that's going to happen, do we want to specifically help those species that are going to be successful?
See, and I would argue that is the major fallacy with the environmentalist movement to this point, is focusing on species. Right. I have been saying, and I will continue to stand up on my soapbox and chant that we should be saving habitats. We should not be saving species. If you save a habitat, you're saving thousands of species. So don't worry about the tree frogs, worry about the rainforest. Don't worry about the pandas, worry about the bamboo forests, because that's where you're doing the real good. Saving an individual species, pulling animals out, starting a breeding program, re-releasing them, that doesn't do anything for them if you're not saving the place where they live. And that actually has a way stronger impact on the health of our planet.
So if there's an open area of Canada that's not really being used for much, can I put a mammoth there?
I was gonna say.
That's I just want to know.
Justin wants to figure out how we can bring back the past.
The mammoth, the mastodon, maybe.
Hey, if the tundra needs help and mammoth poop will help, maybe I'll change my mind. Maybe I'll flip flop on this one.
Well, we have to do the study, so we need them.
Right.
We just need to synthesize mammoth poop for the study.
Oh.
And then following on with the idea of who we should be what should we be conserving, who should we be taking care of, how exactly is it that evolution takes place, which species are going to win out, which are going to be successful, which are not. So there is an experiment that has been ongoing since February 24th, 1988 in the lab of Richard Lenski.
Oh yeah, this is one of those. This is an experiment that was going to show up probably on this show again before too long, as this number continues to get higher, right?
Mm-hmm, exactly. So back in 1988, Dr. Lenski seeded 12 flasks with E. coli, the lab rat of the bacterial world, and then set them up and then gave them only enough nutrients so they could grow, glucose being the main nutrient, but not enough that they would be overfed, just enough to grow. And then every day, I think there's only been like a couple of skipped dates because of natural disasters or something that went on. But I think basically every single day since 1988, someone in the lab has taken a sample of each flask, put them in a new flask with new growth media, and then set them up to grow. And so every 75 days from there on, some of that culture goes in the freezer for analysis. And the conditions basically set them up so that the organisms have enough food to grow, but if they use it all up, then they'll starve overnight. So they have to adapt to these conditions, and this is pressure for evolution. So over the last few decades, 68,113 generations of E. coli bacteria have been born, lived their lives, and moved on. The bacteria have been maintained at 37 degrees with pretty much glucose as the food source, and it's a diminishing supply that adds this selective pressure. And they have seen some results. So the cells have gotten bigger. They use glucose more efficiently, so they don't use it all up so quickly. They grow faster as well. So they're able to grow faster, bigger, while using the glucose better. So this is totally like stronger, better, faster in E. coli. The mutation rate has slowed down, but it hasn't stopped. And there are still some molecular tweaks that are going on. 20,000 generations in, one of the cultures evolved to start eating citrate in addition to glucose, and it is the only line, the only lineage that's evolved to do that. No other lineage has figured that out, has had that mutation to figure it out over the time. And in the time since that 20,000 generations, it's less able. It uses citrate more as the primary source and less with glucose. And so the question that's asked in this Ars Technica article is that the inability to metabolize citrate is one of the things that we use, the characteristics we use to define E. coli. So in the fact that they now metabolize citrate primarily and don't want to really use glucose, does that mean they speciated? Are they E. coli anymore? Was that a mutation that led to a speciation event because of a food source? Questions.
Well, species are just needless labels.
Needless labels. I don't know. It's all
arbitrary.
And then there are 6 of the initial 12 populations that are hypermutators. And they picked up mutations that allowed them to control DNA repair. And so they can get more, or it controls their DNA repair in a different way. So they actually build up more mutations in certain areas of their genomes. And so they have jumps in their genetic diversity that the other species, the other lineages don't, because they're changing, they're fixing the DNA, fixing the mutations in a different way than the other strains did. The other 6 populations are just regular mutators. It's all totally fine. And this is all going on and on, random stochastic mutations leading to diversity. And we potentially have already seen speciation as a result. So 68,000 generations in, and how much longer will this experiment go, and where will it go? Answering questions about how mutations lead to evolution. No, we don't have sentient E. coli yet. That didn't happen.
Just don't let it out. Don't let the smart E. coli out.
Don't connect the E. coli to the internet.
Yeah. God, then we'll get AI E. coli.
I like it. That would be fun.
That's my DJ name.
I could see it. I would
AI E. coli in the house.
in his house, chopping up salad for you. All right.
I've obviously never been to a record listening party, so I'm not catching these references. And instead, I was paying attention to robotic bees.
Uh-huh.
The latest generation of RoboBee from Harvard John A. Poulsen School of Engineering and Applied Sciences can fly, dive into water, swim, and propel itself back out of the water and land. This RoboBee is 1,000 times lighter than any other previous aerial to aquatic robot that has ever existed. So this is very small. This is a millimeter-sized robot that moves in and out of water. Lots of challenges with that. First, water is 1,000 times denser than air, so the wing flapping speed has to be different between air and water. Flapping to frequency is too low. RoboBee can't fly. Too high, the wings snap off in the water. Poor little bee. Researchers found the right rates. It's somewhere around 220 to 300 hertz for flying around the air, and under 13 hertz once it's in the water to move about. There's another problem, though. Getting into the water in the first place. Surface tension is more than 10 times the weight of the RoboBee itself and 3 times its maximum lift. So there's this whole like hitting the water and breaking. That could be a thing. Getting into the water.
It doesn't really look like a bee.
No.
I mean,
You've got to see it fly. Look at the wings. Look at those wings. Those are bee wings. And it's black and yellow.
It's
And
black and yellow.
If you're on the PhysOrg site, there should be a video of it getting out of the water. So there's the other problem is getting out of the water. So they figured out that if they made sharp, pointy things at the bottom, they could break the surface tension of the water and they could drop into the water. Problem was getting back out again. With this bee trying to get out of the water, surface tension, strong as it is, they actually created a pretty amazing technique in this tiny, tiny bee. So they retrofitted RoboBee with 4 buoyant outriggers and a central gas collection chamber. So once the RoboBee swims to the surface, an electrolyte plate in the chamber converts water into oxyhydrogen, which is combustible. So can you see this? Are you screen sharing the video?
Yes.
Look at this amazing leaving the water. Right.
Surface tension is strong. So
Yeah.
this is no mean feat for a small device to be able to escape the forces of surface.
For this little bee, it's 3 times its maximum lift with its wings.
Yeah.
So how did it get out? Well, it created this little pocket of oxyhydrogen and had enough of a spark to fire, to launch, to just break that surface, and then the flapping takes over and he can land perfectly well. Look at that little landing bee, breaking water surface tension and landing like a champ. Like a champ goes, RoboBee.
And next thing you know, we'll have RoboBees on Mars.
Well, one of the kind of fun things that they run
Sure.
into, though, is there's all these different dynamics when you change scale. You know, water tension on a surface becomes a problem, which it wouldn't have in a larger scaled model. Things like the lift of the thing and the surface tension of the water becomes a problem that wouldn't have been a problem before. So they're finding changing the scale, all of the forces that they're used to working with, these engineers, everything is different, and they have to adjust everything. So pretty amazing, fun study there. Don't know how they're actually going to use RoboBee in the future, but if they can make it fly and submerge and land and, you know,
Pollinate the plants on Mars, like Kiki was saying.
we can pollinate plants on Mars. Yeah, because they probably have no problem with radiation. So this may be the best candidate.
Speaking of radiation, space is full of it, and we've been trying to figure out how do things in space get made? Planets. How do they clump up and turn into planets? And so we sent a mission to a comet to kind of help us figure this out. Rosetta went to 67P Gerasimov Chermenko. Well, Churyumov Gerasimenko. There we go. Its nickname is Churi. Churi. I like that much better. So the Rosetta mission, the lander actually got stuck for a while, wasn't able to get data back, but they did get some data back. And so a research team out of Technische Universität Braunschweig, Germany, Jürgen Blum and his team have published in the monthly notices of the Royal Astronomical Society on their study of particulate matter in the comet, the surface structure of the comet. And what they have determined is that based on the surface structure of the comet, what they were able to measure that was there is that we could get rid of a whole bunch of hypotheses about how planets are formed, because based on what they saw on Churi, all you have are dust and ice particles that are attracted to each other and that get stuck to each other, that the uppermost layers are irradiated by the sun. Radiation, there's that link right there. So that there's no ice on the surface, but beneath the surface, there's a cold mix of dust particles and ice that eventually, as they accumulate together, are held together initially by molecular forces, but then later it becomes gravitational. As they get bigger and bigger, the clumps get bigger and bigger. And the evidence from Rosetta is that our idea that the dust and ice rings, or the particulate matter that surrounds a young star, over time, it just starts, they bump into each other. Dust and ice, it bumps into it, and another piece bumps in, another piece bumps in, and it aggregates. And eventually you have something the size of a comet. Maybe that comet bumps into something bigger. You have aggregation, and that is how this study suggests that planets form.
Yeah, nice confirmation. It's also
confirmation of the idea, right?
It's the best idea we got so far.
Yeah.
I don't know if it was the Itokawa, the Itokawa asteroid that Hayabusa was trying to land on. But it had it was like a gravelly. It was described as not a completely solid rocky surface, but like the whole thing was made out of clumped gravel. That a lot of these asteroids out there might be big clumps of gravel, like all these smaller bits that haven't completely coagulated until they need to get bigger. They need a larger mass. They need to run into a few other things to start to get that compression going.
Yeah, and so
I'll start small.
Yeah, it starts small. And so the researcher, Bloom, he says, although it sounds very dramatic, it's actually a gentle process in which the dust agglomerates are not destroyed but are combined into a larger body with an even greater gravitational attraction. The accumulation of the dust agglomerates into a coherent body is virtually the birth of the comet. Yeah, and he says that this is a big step in the planet formation model. So thank you, Rosetta. Thank you for that. Got another story, Justin?
Yeah, this is a fun one. People with this is Quotey voice. People with efficient brains may have too much brain capacity to stop their minds from wandering, says Eric Schumacher, Georgia Tech Associate Psychology Professor. Schumacher and students and colleagues, including lead co-author Christine Goodwin, measured the brain patterns of more than 100 individuals in MRI machines. Participants were instructed to focus on a stationary fixation point for 5 minutes, and they used that data to identify which parts of the brain were working in unison. They had also filled out one of those little flyers where they ask you lots of questions. Lots of questions, hiding the one that was probably the most important. Do you daydream frequently? Was the question that they were really focused on. And interestingly, it turns out that those who said that they daydream frequently scored higher on intellectual and creative ability and had more efficient brain systems measured in the MRI machine, leading us to the overall conclusion that if your mind wanders in a boring meeting, it's because you're smart, not because you can't pay attention.
I'm sorry, what were you saying?
Exactly. Oh, except for the smart ones in this, they were able to pick up key points of data and return to the meeting as if their minds had not wandered. That was
yeah, I was totally listening. I heard everything you said. Something about daydreaming being good. I don't know.
Yeah, they say, how can you tell if your brain is efficient? One clue is that you can zone in and out of conversation or tasks when appropriate, then naturally tune back in without missing important points or steps.
That sounds like multitasking, but where one of the things you're doing is not important.
It's just daydreaming, yeah. Which might be the meeting or it might be the daydreaming.
Yeah,
it just all depends.
Yeah. Sometimes it's both or neither.
Well, in terms of education, I mean, this has implications for those students who are who daydream, who end up somewhere else in class, who maybe are the class clowns doing other things, coming up with ways to entertain themselves.
Yeah.
Sometimes these kids are the kids who have figured it out. And so as a teacher, as an educator, how do you keep their minds active? How do you keep them I mean, daydreaming is great, but how do you keep them engaged in the class? And that is the ongoing question.
Yeah, Schumacher here also says higher efficiency means more capacity to think, and the brain may wander when performing easy tasks. So it may be, if your mind is wandering, it may be not just might not just be signaling to your boss that you're bored with their conversation, but that you just get it, and it didn't really need to take an hour and a half to get to this information.
Do we still need to be talking about this?
Yeah, most business meetings are absolutely essential down to every minute. So I don't know what you could be talking about.
All right, so that we get down to the business in the last minutes that we have here, it's time to do some quick, I don't know how quick these are going to be, but this is what we call it. This is our quick news segment that sometimes ends up being a longer discussion depending on the story. So interesting study out of University of California, Berkeley. Researchers got funding from the National Institute of Health and Heising-Simons Foundation to investigate gun violence in California. Published in Annals of Internal Medicine, they looked at gun shows in Nevada and their relation to gun violence in California. They found that there was a almost 70% increase in deaths and injuries from firearms in communities in California within convenient driving distance of Nevada gun shows.
Huh.
So for
Very
a couple of weeks around gun shows happening in Nevada, places in California experienced a spike in deaths and injuries due to gun violence.
Well, it would be great if we could take that from correlation to causation by figuring out if the people responsible for that gun violence attended the gun show.
Right. So we can't do that exactly, but that would be the next question. But what they were looking at is the idea that California is very restrictive in its gun laws. Nevada is not. And so people from California probably tend to go to Nevada, purchase guns, trade guns, come home, and do it under Nevada law in Nevada, and then drive the weapons home.
Technically, it's not that easy. Actually, the thing that surprises me, though, about this study isn't that they found this correlation, but that the National Institute of Health funded it. Study.
Yeah.
That's like...
Yeah, that's a step in the right direction for sure. But it's
researchers. Go ahead.
It's just frustrating because I feel like we could be doing multi levels of this study at the same time, and it would always point to the same thing. So, how many times do we have to show that guns cause violence?
One more.
One more time. Just one more.
Maybe two.
Okay.
3 would really be building a case. Another 17 of them, and I think we're getting close to a discussion.
Right.
Too soon to have that conversation.
Oh, yeah,
It's going to take 30 more times before we're at a point where, but it's probably, again, too soon after those 30. What we really need to do...
Yep.
So the researchers say the study suggests that travel to less restrictive states may threaten the effectiveness of firearm laws within California. When a less restrictive state is next to a state that is more restrictive, there may be spillover effects. More research is needed to know for certain. So maybe it'd be nice if there would be more funding for this kind of research, just to know for certain.
Yeah.
That would be awesome. And then... I have a couple more. I'm going to run through them. Researchers at Northwestern University have been studying RNA interference. And so RNA interference is this concept in which we use small segments of RNA to bind with and interfere with gene activity. They have been searching for molecules, small double-stranded RNA molecules that they think are an ancient, not used anymore because we have an immune system that has evolved differently, but an ancient kill switch to prevent cancer. And so he's been looking for this. The researchers have been looking. Peter, this researcher, Marcus Peter at Northwestern, he's been looking at this for several years, and he says, we think this is how multicellular organisms eliminated cancer before the development of the adaptive immune system, which is about 500 million years old. It could be a fail-safe that forces rogue cells to commit suicide, and we believe it is active in every cell, protecting us from cancer. And so the research that they have published recently, they've done a phase 1 and a phase 2 trial. And they've done that's a different study that I was going to talk about next. This one, they have actually discovered some RNA sequences that bind to DNA that is related to cell death. And so this RNA assassin molecule triggers this mechanism that Marcus Peter calls DIES, death induced by survival gene elimination. It's pretty awesome, right?
And that's the name of the band I'll be spinning all night. So,
right. And so they have tested this treatment in mice, DIES treatment, and they delivered the assassin molecule via nanoparticles to mice with human ovarian cancer. The treatment reduced the tumor growth. There was no toxicity. And the tumors did not develop resistance to this treatment. And so this is something that will specifically address solid cancer treatment, so tumors that form in people's body. But what is really exciting is that they're looking at a small number of molecules that can tap into this metabolic kill switch that only kills cancer cells. Really interesting.
Metabolic kill switch.
A metabolic kill switch. Yes. There was a quote from the lead author, Marcus Peter, describing like how effective this possibly is, these RNA molecules eliminating all these survival genes at once. And he says, it's like committing suicide by stabbing yourself, shooting yourself, and jumping off a building all at the same time.
Sounds like overkill.
But that's good in the sense of if this is only happening to cancer cells.
Yeah.
That's great.
Yep.
It's great. And then my final story was related to vampires. The vampires that the elderly are going to be as research moves forward. So the new research now is looking at stem cells taken from younger individuals. They took bone marrow to get mesenchymal stem cells from donors between 20 to 45 years old. And they have used them in phase 1 and phase 2 trials to test the effectiveness of these mesenchymal stem cells on treating frailty in the elderly. And to date, frailty, go outside, get some exercise, go for a walk, get moving, do something, maybe your diet can help. There's not much that we can do for the frailty that comes from the degeneration of your muscles and your body as aging occurs. And so the patients in these trials had an average age of 76 years old. They were infused with these young people's stem cells. And it helped. I mean, it's a very small sample size. The phase 2, they looked at 15 frail patients. 6 months later, all the patients had improved fitness outcomes. All their tumor necrosis factor levels were better, and their overall quality of life was good. And there were no negative or adverse health effects. And they did a randomized double-blind study with a placebo group, which is a big deal. You need that randomized double-blinding with a placebo to make sure that you're not confounding factors and adding bias in anywhere. No adverse effects. Physical improvements noted by the researchers were remarkable. So, publishing in the journals of gerontology, these researchers say they're going to be moving forward into an expanded phase 2B trial. They're looking for 120 subjects across 10 locations, and then they will move to phase 3 trials that will look at thousands of individuals to potentially lead to public approval. So far, we have a small group of people that nothing bad happened, and it was a positive effect.
So this definitely sounds like vampires, for real.
Right? Oh
yeah.
my God.
But there's part of me that's like, great, this is going to get me closer to 200, baby. I'm on board.
Yeah, if they're doing the research now, Blair, you're set.
I'm going to be set.
You're set. You're that's so much younger than I am.
Oh my God, not even that much.
But just so you know, that's also going to raise the minimum age for collecting Social Security to 145.
That's right.
It's Justin. Social Security. What are you? That's not going to be around in 10 years.
Oh, no. That's going to disappear over the infinite horizon is when that's calculated to stop working, which, by the way, When you do calculations over an infinite horizon, that means it's never today. Yeah.
Right.
It'll be fine.
Real quick before we go, do you want to hear about spider venom?
I do.
Okay,
VR spider venom.
VR spider venom.
Yes, I do.
What?
Yes, a new virtual reality game highlights the benefits of spider venom in pain management. UEA's School of Pharmacy, University of East Anglia Pharmacy and School of Computing Sciences is looking at ongoing research into the potential of spider venom to stop pain and cure chronic illness. So this VR game makes a view of your brain, submerges players into the world of the brain, where they learn how pain is processed in the brain, why we sense pain, and how spider venom has the ability to alleviate it. It teaches us about the functions of cells and proteins that sit on these cells that are part of pain treatment. And in the game, virtual spiders release their venom when you click on them. Then the player has to quickly work out which of the spider venoms can shut down pain, and it ends when the player identifies the perfect venom to block pain-causing proteins, which ends with the words, "Pain over."
Of course it does.
Yeah. So it is debuting this Friday, the 27th of October at the Norwich Science Festival, and it is called Bug Off Pain.
Bug Off Pain.
Bug Off Pain. If you have
right.
VR systems, you will be able to download it from bugoffpain.com starting this Friday. Check it out.
Bug Off Pain.
You can explore your brain, pain, and spider venom.
I wish I had VR now.
So spiders in your brain.
I need to
Good thing.
get some cardboard and make some Google goggles.
Yes.
Yeah,
Yes.
right.
Who thought spiders in your brain would be good?
Spiders in my brain. Oh my gosh.
There we go.
Spiders in my brain. You know, I think some people get spiders in their brain and it's not because of the venom.
I don't want to talk about that because every night I cover up my ears because I don't want spiders going in my ears.
Nuh-uh.
You're not.
No,
You're
I
not.
used to do that when I was a child, though.
Oh my gosh.
Every night I would cover a sheet over my ear because I wouldn't want spiders in my ears and brain.
Yeah, I had a really quick conversation with my daughter about spiders where she was saying she didn't like spiders. And she says these things every once in a while, but
Just show her jumping spiders.
well, I go, well, like, what's worse, mosquitoes or spiders? And she hates getting a mosquito bite. So she went, mosquitoes. And I was like, well, spiders eat mosquitoes. And if it wasn't for spiders, there'd be more mosquitoes. And now she loves spiders. She's like, there's a spider, but we're not going to touch it, but we're not going to squish it. I'm like, yeah, we're just
There we go.
Good.
Yeah. We're not going to touch it. We're just going to leave it alone unless it's on my bed.
And it's one of those things
No
too,
spiders on the bed.
They picked this up other places. Like she says, mice are gross, right? I'm like, where did you? Because should we go down to the pet store and she wants to like hold the mice and have them run around on her hand, and she thinks that they come up on the cage and they're like wanting to talk to her, and she's like really loves them. But somewhere she heard that mice are gross, so now she says this. I'm like, so you don't want to go see the mice down at the store? And she's like, no, let's do it.
Yeah, so spiders also have venom that
spiders.
can cure pain.
Yeah.
Pain
Dr.
curing spider. That's right. I want a doctor named Dr. Spider.
Dr.
Yeah.
Spider.
And spiders, they're
Give
not
me-
closely related to us, and a lot of them that can cure pain are not from here. So take that, Dr. Guy.
Going back to that conservation conversation.
I got a
Dr.
wooden spoon with your
Guy.
name on it, Dr. Guy.
But
Take
Dr. Guy, just so you know, Blair does agree with you about pandas.
To an
Maybe-
extent.
Maybe we should invite him to talk with us on our show at some point.
Oh man.
That would be one of those shows where I'd just be like this.
Yeah. Be sure to do that virtually. I don't want to be in the same room.
That would be so fun. Oh my
Things get
gosh.
difficult.
I want to see Blair go aggro. But you know what I really want to see right now? I want to see our scroll. I want to see our roller for our Patreon sponsors, because we've gotten to the end of another show. We've made it all the way to the end. And I just want to thank everyone for listening, for watching. All our chat room, thank you for the vibrant conversation in the chat room. Always keeps me smiling. And to Fada and Identity 4 and Brandon for helping out, all the things you do, keeping the show going. Love it. Couldn't do this without you. And our Patreon sponsors, for sure. You ready? I got a new scroll. I'm going to try and read the names in a different order.
Is it spooky?
No, it's just the normal way. Thank you to our Patreon sponsors: Honey Moss, Aaron Luthen, Adam Mishkan, Alec Doty, Alex Wilson, Andy Groh, Arlene Moss, Artyom, Ben Rothig, Bill Kersey, Bob Calder, Braxton Howard, Brendan Minish, Brian Hedrick, Brian Condren, Brian Hone, Bruce Cordell. Oops, and I reset it. I don't know how I did that one.
It wasn't until you put it alphabetical that I realized how many Brians there are.
There's a lot of Brians, and now it's not loading, and an error occurred, and you're gonna see my drive because it's all open. Yeah, we're gonna keep going. Patreon. Honey Moss, Aaron Luthen, Adam Mishkan, Alec Doty, Alex Wilson, Andy Groh, Arlene Moss, Artyom, Ben Rothig, Bill Kersey, Bob Calder, Braxton Howard, Brendan Minish, Brian Hedrick, Brian Condren, Brian Hone, Bruce Cordell, Byron Lee, Charlene Henry, Christopher Dreyer, Christopher Rappin, Columbo Ahmed, Craig Porter, Dale Bryant, Dana Pearson, Daniel Garcia, Darwin Hannon, Daryl, Dave Neighbor, Dave Wilkinson, David Friedel, David Simmerly, David Wiley, Donald Trump the Dubious, Dougal Campbell, E.O., Edward Dyer, Emma Grenier, Eric Knapp, Eric Wolf, Felix Alvarez, Flying Out, Gary S., Gerald Sorrells, G. Burton Lattimore, Gerald Onyango, Greg Guthman, Greg Riley, Harun Sarang, Hexator, Howard Tan, Illuma Lama, Jacqueline Boyster, Jake Jones, James Dobson, James Randall, James Dozier, Jason Olds, Jason Roberts, Jason Schneiderman, Jean Tellier, Jim Drapeau, Joe Wheeler, John Atwood, John Crocker, John Gridley, John Ratnaswamy, Keith Corsell, Ken Hayes, Kevin Parachan, Kevin Railsback, Ken Seneyev, Volkova, Kurt Larson, Larry Garcia, Layla, Louis Smith, Mark Mazzaros, Marjorie Mark, Marshall Clark, Matt Sutter, Matthew Litwin, Mitch Neves, Maura Cowbell, Mountain Sloth, Nathan Greco, Orly Radio, Patrick Cohn, Paul Staten, Paul Disney, Phil DeJoe, Philip Shane, Randy Mizuko, Richard Hendricks, Richard Odermis, Richard Porter, Rick Ramus, Richard Aston, Rodney, Rudy Garcia, Salgood Sam, Shiwata, Sir Fricadelic, Stefan Insom, Steve DeVell, Steve Lessman, Steve Mashinsky, The Harden Family, Todd Northcutt, Tony Steele, Tyler Harrison, Tyron Fong, Trainer 84, and Ulysses Adkins.
There were new ones.
Yeah.
So many new names. I love being able to read this and have it updated every month. So thank you all, our new Patreon sponsors, our old Patreon sponsors. Thank you so much. Thank you for supporting us on Patreon. If you are interested in supporting us on Patreon, you can find information at patreon.com/thisweekinscience. And remember, you can help us out simply by telling your friends about TWIS. Also, remember, if you go to twis.org, you can order our 2018 calendar if you are so interested. Twis.org. You can pre-order the calendar. The link is there. And on next week's show, November 1st, once again, we will be back live, 8 p.m. Pacific time, Wednesday on twis.org/live. And you can watch and join our chat room online. But if you don't make it, that's okay. It's all right. We're all archived various places. You can go to twis.org. You can go to twis.org/YouTube. You can go to facebook.com/thisweekinscience. We're all over the place, man. You can find us.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can look for TWIS, the number 4, Droid app in the Android marketplace, or simply This Week in Science in anything Apple marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org, where you can also make comments and start conversations with hosts and other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIScience, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
And we will be back here next week, la prochaine semaine. And we hope that you will join us again for more fantastic science news.
Arr, and if you've learned anything from the show, remember,
Sistras, my science babies, is all in your head.
Happy Halloween!
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma. Got the eye. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you learned anything from the words that we said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Und maintenant, nous allons à le post-show.
Oui,
Oui,
je suis prête.
je suis prête pour le post-show.
Oui.
Nous avons fait le show.
Oui.
Et maintenant, nous avons le post-show.
Un petit post-show?
Un petit.
Ou
Un
un
petit.
grand post-show?
Je peux fais le packing pour le voyage à San Francisco demain. Ouais.
Oui, il faut que tu fais le packing.
Fais le packing. J'essaie du packing, mais c'est difficile.
Oui.
Parce que il est chaud, il fait chaud.
Oui, c'est très chaud. C'est
Je suis confuse.
80 c'est 80 degrés Fahrenheit aujourd'hui.
C'est dommage.
Oui, c'est dommage.
C'est dommage. Ce n'est pas le San Francisco de...
C'est le garbage.
All right, everyone out there. We are using our poor French to have a conversation, but we actually had a conversation there.
Yeah, we did.
We did. It was understandable to
I have
ourselves.
a French minor. That's the most I've used it in probably 5 years.
Yeah, back a long time ago when I was an undergraduate, I wanted to get a French minor, but then I realized that they'd make me write in French a lot, and I just wanted to speak it.
Yeah, I wrote a 20-page paper.
Oh, geez.
In French.
Yeah. That was why I didn't get a minor in French.
It was not easy. It was not easy.
No. Yeah, I was like, no, I'm not going to do that. I think I just want to speak it. And so then I went to France for a little while, and I tried to. Have I told you this story? I tell people this story. I went to France and I tried to buy a book at a bookstore. And I was so excited. And I went to the woman who was selling the books at the bookstore. I said, "Avez-vous le Petit Prince?" And she said, "Would you like to speak English?"
I know. They like to do that there. It makes me really sad. Although
you're so mean.
The last time I was in France, I was trying to return a rental car, and I could not find the place to return my rental car in the middle of Paris. And I ended up pulling into a rental car return, but the wrong company. And so the guy there didn't speak any English. And so I had to try.
You had to, yeah.
I had to do it. And I had to get directions. I'd explain. I had like an Avis rental car that I had to return. And so I had to explain that I needed to get to the Avis rental return. And I had to get directions on how to drive there. And it worked out. It was clunky as all get out, but it worked out.
Et prochaine, vous traînez à la droite.
À la droite.
À la gauche.
À la gauche. Yeah.
Gauche. À la gauche.
Oh my gosh. But it'd be like at the street light you make a right, and yeah, I don't even remember how to say that now. Anyway,
my favorite French in TV is from I don't know if anyone's gonna get this. It's from a cartoon. Mais je suis un homme. Je ne suis pas un oiseau.
Oh, that sounds very familiar. I'm a man, I'm not a bird. What is that from?
It's from The Tick.
Yes.
Where he was turned into a bluebird and put in a cage, and he could only speak high school French.
Oh my God. That's great.
That's one of my favorite things ever.
I'm gonna have to look that up.
Oh my God, one of the best scenes ever.
I remember in high school doing a translation of Alouette and being horrified about what that song is actually about.
Once I learned what the words actually meant.
Yeah.
What
They're
does it
preparing
say?
a bird to cook it, but they're like talking about, you know, cute little
Alouette.
bird, yeah, pretty
Yeah.
little bird.
Gentil, gentle, gentil, beautiful, pretty, gentil,
Yep, very
pretty.
And-
Alouette.
Alouette.
Gentil plumeret.
Right. So they're plucking the bird,
Plumes.
and then they say, "Oh, I pluck the beak. You pluck the beak. I pluck the beak. Oh, little bird, pretty little bird, I pluck you." Right. So then they say, "Oh, next I pluck the feet." It's like piece by piece by piece. They're prepping this bird to be cooked. But the way the song sounds, you think that they're just singing about a cute little bird in a tree.
Mm-hmm.
So
Things
it's kind
you
of
learn.
it's pretty upsetting. But I guess we have our own children's songs that are pretty gross, like Ring Around the Rosie and stuff. So
Je te plumerai la tête, je te plumerai la tête.
pull the whole head off.
À la tête, alouette, alouette. Oh, that's right. I will pluck your head.
Yeah, pluck your head off.
Oh my God. Je te plumerai le bec, je te plumerai le bec.
Yeah.
Je te plumerai les yeux, je te plumerai les yeux.
Pull the eyes out. For sure.
Oh my God, it's so. I think pluck your eyes out is the.
That's definitely that one in the feet. Pulling the feet off really bothered me. I was like, ew.
Je te plumerai les pattes, je te plumerai les pattes. Le queue, les ailes, le dos. Oh no, no, no.
Ugh, it's hot. I'm wearing a chef's coat and it's hot.
You're wearing a chef's coat.
Can't.
French
falling apart.
chef with the French
Yeah.
bread.
Ugh,
Post show, Blair can't do it anymore.
it's too much. It's really hot. I closed the sliding door so the whole neighborhood didn't have to hear me do TWIS.
And so it's warm in the house with the no air conditioning and the 80-degree day.
It was 90 yesterday.
Oh my gosh. It was like 108 in either Santa Barbara or San Luis
San
Obispo.
Diego, I think.
Was it San Diego?
San
Some-
Diego
See,
had
and
the
they're
heat
not-
warning.
They're not supposed to get the Indian summer. We're supposed to get the Indian summer. That's fine. Like this
Yeah.
is supposed to be our summer. But we also had a hot summer, so that also is like not great. But yeah, they're not supposed to have that now. They're supposed to be cooling down for fall right now. And they're not.
My cat's little paws are underneath the
Is it like
door.
this?
She's like sliding them under the door and like looking for something. Yeah, that's really cute. She wants to grab something. She wants in. Hi, Stella. Hi, Stella. Hi, Stella. It's kitty time. She's like, I know it's the end of the show. This is the time where I get to get pets. After show, I get pets.
So did you guys see my my proof of the calendar?
Yes.
I have to finish the
we need to make sure the holidays are all right.
right. So I'm working on that.
And if there are any more
this weekend.
holidays that people might think of.
Yeah. So I have our I was like going through it and I didn't screen share. That was dumb. So I'm going through our document from last year and fact checking all of the current of the 2018 dates because sometimes things change. So I have to go through and make sure all those are correct. I also have another fun list of dates that I'm going to look at
to see about adding. Nice, good.
And
good.
yeah, and then it'll be ready to go. So the dates might switch too depending on which holidays or which days, but so right now there's the cover and then we have the anaconda.
So
into shrimp.
Hold on 1 second.
Squirrel.
So we had our TWISoween.
Tiger. coral reef, dodo, tiger salamandre, leopard shark,
Let's see.
flamboyant cuttlefish, tardigrade, and pangolin.
So looking a month ahead. Oh, you know what? We had TWISoween scheduled for next week.
We did?
Uh-huh.
Oh no. Oh man. We messed it up. Oh well, we did it early.
It makes sense.
This
Yeah.
makes more
We
sense.
should always do it before the
Yeah.
actual holiday.
Yeah.
Sorry, people with calendars.
Whoopsie.
I have a calendar. I just listened to Blair.
I
Blair said it
know.
was
I
this
have a
week, and I was
calendar
like,
right there.
I was like, okay, it's this week. All right.
I had it in my phone as this week, which is funny because I pull from my phone calendar for that calendar. So I effed that up somehow.
But as we come into November, it's not on the calendar, but November 6th, 5th, 6th, 7th, we're in Denver.
Yeah.
So that's coming right
so
up.
soon.
Justin, have you registered for the conference yet?
Yes.
You did. Okay, good.
We have plane tickets because I didn't do that.
I got us plane tickets.
Yeah, okay.
Yeah, we do.
It
We
was
have
today.
I thought I mailed you that stuff, emailed you that stuff.
Yeah, I'm sure you did. But just today, like at work, I was walking around and I was like, I didn't book a flight.
I did.
think Kiki did.
I did it
It's
for
been a
us
while.
all.
That's great.
It's all taken care of. Their travel people took care of it. And
Yeah.
I really wanted one airline and they didn't do it. So now I'm not going to get
Right.
miles on my plan, but that's okay. You will because it's your airline.
Oh, it's my airline. Oh, great. It's the best airline.
So we have conference and live show on the 6th. I'd like to do a Denver meetup on the
Great.
5th or 6th. We could do the 5th or the 6th, but I'd like to do a meetup.
Well, we arrive at 2:17 p.m. on Sunday.
Mhm.
So, or at least I do. When do you arrive?
I mean, maybe it would be fun to do the meetup on, yeah, we all arrive around the same time. That's what the
Okay.
travel agent,
Okay,
Did.
so we're traveling from the airport together.
yes, to
Okay.
the hotel.
I'm into doing it the night before, potentially.
Okay.
I don't know. Whatever you want.
Cool.
We could.
All right,
we-
because it's like a week and a half out. And so I'm
Yeah.
thinking, or 2 weeks out, exact, a week and a half out.
unless you
week
think
and a
we
half.
need to be prepping that first night at all.
Yeah, we might want to do prepping and not drink a little.
Okay,
I'm
yeah,
gonna
that's
be
a
doing
good. That's,
pimping the whole
Yep.
time.
And it would be
So that
like
I'm-
if we did it the
fabulous.
6th.
yeah.
And we
Then
could-
the show's over already. So
And
we
so
can-
then now we have to answer a question though. If we do it on the 6th,
Uh-huh.
is that going to be our show for that week? Because I can record it. That's what we're going to do. We can record it and we can also make it live.
I say no.
And will
Okay.
that be our show for that week?
I say no.
I say.
Does it come home Tuesday and then Wednesday?
I see 3 options. No, 2 options.
Will we put out 2 shows that week?
So one option is we record it and we do a live intro to the recording and we find a way to air the recording on Wednesday. Just so we can be like, hi, everyone. We're going to play our show. Just so, loyal visitors will still have a greeting from us on Wednesday. But also we could potentially borrow stories from Tuesday or Monday for the Wednesday show.
Yeah. I mean, the reason I'm for 2 shows is because I don't think this will be one of the jet laggy ones because we're not going really far.
We're not going that far. Yeah.
So I don't think our recovery time will be impacted. I don't know. And we're going to be very specifically sort of interviewing and bugging out with the entomology.
It's going to be a very buggy show. very bug
so-
focused.
You're so funny.
Well, so I mean, like, there's going to be a lot of other stories that we'll probably want to have brought for that week that will then.
Well, you guys.
Right, I know, Blair. I know. Like, I was almost
I'm
like,
probably gonna double dip.
I know, but I was also like, why are we, me and Kiki going? Like, Blair could do the whole show.
Right.
Get out of here.
She
You
doesn't
don't
need
want
us
to
for
give
this
me
one.
that idea. You don't want me with those ideas.
She doesn't need us for this one at all. This is her, like, I'm gonna have to like find insecty stories. I don't even
This
know
is
what
my
those
house
are.
of wheels. That's true,
Yes.
but it's just not TWIS without you guys.
Yeah, no, we're still going. 'Cause
Yeah, you better come.
We want to see Denver.
Otherwise, I'm just gonna show up and run around looking at things.
But yeah, this is I expect the Blair's Animal Corner to dominate the airtime on this one.
That would be
Yeah. All right. So maybe two. So maybe 2 episodes that week. All right. If
Yeah,
Justin, if you're down, Blair, if you're down, I mean, we can do that.
I mean, I never have anything to do on Wednesdays but this. So if we don't have TWIS, I will be going to bed at 6 p.m., which would also be fine. Heavens
Awesome.
knows I need it. But I am happy. I am happy to do a second TWIS.
I don't know what you guys mean needing sleep. I don't know. Right.
Oh, there it is.
Anyway, okay, so there's that. So we can do 2 shows that week. And then Thanksgiving. That Wednesday, it's not Thanksgiving. It's the night before Thanksgiving. Are
We
we
said
going
we
to-
would do a show, right?
Yes, we always kind of do that. Are we going to do that?
Yeah. Sure. Justin?
Yeah. Oh, yeah. Like that's a fine. I'm not doing anything Thanksgiving Eve.
Mm-hmm.
Cool. And then all the other days, like we're not on New Year's or Christmas in December, so we're good for that.
Excellent.
Yup. The holidays are coming, you guys.
Oh my gosh, I can't believe it. Yeah. It's crazy.
I know. I'm not ready for it. We can get ready for it though.
I'm not
I'm gonna buy everybody TWIS mugs.
Yeah.
And you get a TWIS mug. And you get a TWIS
And
mug.
you get a mammoth pillow. And you get a mammoth
I
pillow.
love those pillows. They're so great. You get a mammoth pillow.
I got a bear pillow for my mom. She likes it.
That's awesome.
Mhm.
You better like it. You made it ish.
Yeah.
Right.
I toast made it.
Yeah. Okay. Let's see. I'm coming to San Francisco.
Yeah. So you're in town for the
I'm-
festive weekend.
Yeah. And I think I was looking at the conference schedule and I think I'm going to I don't think I'm doing anything Saturday night, and I'm going to completely blow off Sunday of the conference. And then we leave at 3 on Monday.
So there's a really cool event happening Sunday night
Okay. What is it?
called Bowling for Rhinos.
Oh.
So, and this
That's
is
fun.
to anyone else listening who's in the San Francisco Bay Area. It's a fundraiser for rhino conservation, but according to Dr. Guy, who cares? Anyway,
Dr. Guy.
Dr. Dude. So anyway, yeah. So it's bowling, pizza, sodas, and then we also have a silent auction with lots of animal like art and spa packages and signed like sports memorabilia and all sorts of crazy stuff. But it's it's a fundraiser for rhinos, and we also have a costume contest because it's bowling for rhinos. And yeah, so it's at the Presidio Bowling Center. And on my Facebook page, there's links to the Eventbrite, or you can just show up and pay at the door. It's on Sunday, the 28th.
Awesome.
So There's that, and then I might be available during the day on Sunday a little bit. But
What time does it start?
6.
Okay. So it's not a kid thing.
Kids come. We can put up bumpers.
Sweet.
Yeah.
That would be awesome.
Yeah.
I got a kid who's going to be with me.
Yeah. And he'll be in a room with a bunch of zookeepers dressed up for Halloween.
Oh my goodness, that would be so fun.
So
I love that. Oh, and maybe Dave Robinson is going to do it too. Sunday.
Wow.
Oh,
Here.
Ed, you're so great. You are.
Oh.
Ed already got the link up there.
Jeez, I just have to do that. Ed.
So fast. Ed, you're
Gosh.
fantastic.
So speedy.
So speedy. Yeah, Jackson Fly. Justin, you need to watch Orphan Black. It's so good.
Okay.
So good.
Yeah, and if anyone shows up, I'll be happy to sign things or whatever. I don't know.
You should. That would be awesome.
Yeah.
Bowling for rhinos.
I'll be in costume, not in this costume.
Different
in my
one.
actual Halloween costume.
I know it's a weekend. That it's a celebratory Halloween weekend. My costume, I guess I can pack in. It's just pants and a jacket.
Yeah, there you
go. And makeup. Yeah. My friends have makeup. I bet I could borrow makeup and make it work.
You could also just buy it at Walgreens for like $3.
Yeah.
I might have to get more mustaches.
Yeah, because I wasn't going to bring a costume, but maybe I should.
Yeah, I think I might run out of mustaches by the time the weekend's over.
A mustachio? A question.
What is your question?
Is it time for us to be done now?
Yeah, I think so. It's time for bed. I'm throwing mustaches. That definitely means it's time for bed.
Yeah. My sisters.
I don't like this one so much. It makes you look like you have a big nose.
Oh my gosh, you guys, I went and saw Depeche Mode this week.
You did?
I went and saw Depeche Mode. It took me back to high school, except for some of their new songs, which are good. They're very politically oriented. Like one song is like, "Where's the revolution?" You know, and it's like all these images of fists up on the screen. Like they want people to be fight against the machine.
Right.
But Dave Gahan, lead singer of Depeche Mode, he definitely looks like John Waters these days.
Ooh.
He's got like that little tiny mustache.
I love John Waters.
Yeah, so he's got that small little black mustache. And I was like, Dave Gahan is really, and he's got the kind of long, beakish nose like John Waters. And I was just like, you're working the John Waters angle, Mr. Depeche Mode.
Yeah, he's going for it.
Going for it. All right, everyone out there. It's another week, another show. It was TWISoween. And that was fun. That was super fun. We did it a week early. Sorry, everybody. If you...
Whoops.
If you were really holding back.
I just love Halloween, you guys. I just love it.
Halloween was coming.
I have 3 costumes this year.
I need to have more costumes. I do.
I'm glad we did this drive because I'm missing part of my costume.
Yeah.
And
now you know what you're missing and you need to find
what
it.
I need to go do.
So this is one. For Boo at the Zoo on Saturday, I'm going to be the safari guy from last year's TWISoween.
Oh, yeah.
Yeah.
And then I have a very special costume for my actual like Halloween parties that I am saving.
You go to parties.
I go to parties.
Don't be a party pooper.
Oh.
Because who poops at parties?
Only party poopers.
Okay, I'm gonna say goodnight, Justin.
Good night, Justin. Oh no, wait, I'm doing it wrong.
I'm just procrastinating because I don't want to pack. Because I hate packing.
Yeah, Ed, I'll be sure not to let any spiders crawl in my ear tonight. Thank you.
I'm gonna wear these suckers all night long.
Yeah.
I'm gonna take mine off for my hat, which I can't reach now. But somebody said I'm supposed to turn it sideways. I know, but this one doesn't turn sideways. It's a pointy part.
Uh-oh.
Pirate hat forward.
It's all the flour.
Say goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin.
Okay, good night, Kiki.
My name is Helena.
Good night, Elena.
Good night, sisters.
Je suis Claudette.
Don't lose my babies. My science babies.
Au revoir.
Good night.
Bon appétit.
Happy TWISoween, everyone.